In this program, we're going to discover the various methods of reproduction that exist in nature. We will then broach two subjects from a specifically human angle: contraception and new reproduction techniques. The most conventional method for human beings to reproduce is to have sexual intercourse. However, today, techniques like in vitro fertilization have allowed for conception not necessarily related to intercourse. This artificial means of reproduction remains sexual in that it involves the union of a female cell and a male cell. The great majority of living things reproduce sexually, but there are other means of reproduction. One of the main preoccupations of all forms of life, from unicellular beings to human beings, is to produce descendants, to perpetuate life. That, above all, is the purpose of sexuality. Most living things are born from the meeting of two sexual cells of different origins. However, in some lower animal and plant species, reproduction can be achieved without sexual occurrence of any kind. Called asexual reproduction, two processes are necessary: budding and fission. In budding, a small segment of an individual separates to constitute a second individual that will become a carbon copy of the first. A number of unicellular organisms and primitive animals, such as corals, sponges, and sea anemones, reproduce by budding. The second process is fission. Fission is used by most bacteria and protozoa. An individual divides into two new identical individuals. These two types of asexual reproduction result in the production of individuals genetically identical to their parents. However, these organisms face a certain danger. If unfavorable environmental changes occur, all the individuals will be similarly affected, and in all likelihood, none will survive. Conversely, sexuality introduces variability between individuals. Sexual reproduction is characterized by a particular phenomenon called fertilization. Fertilization involves the fusion of two cells, one male and one female. That fusion involves a close blending of the genetic material from the male parent and the genetic material from the female parent. The egg that results from this fusion will become an individual and is then equipped with new and unique genetic potential. By creating diversity between the individuals of the same species, sexuality may improve a species and enhance its chances of survival. Fertilization, which is the most important event in sexual reproduction, usually involves the meeting of two beings of the opposite sex. Usually. But there are other organisms either fixed or slow in their movements, like earthworms and soft water snails, that have both male and female reproductive organs. They are said to be hermaphrodites. Several flowered plants also produce male and female sexual cells. In spite of that anatomical advantage, most hermaphrodites practice cross-fertilization, meaning the fertilization occurs following interaction between two different partners. Also, the fertilization has to take place in a liquid environment where the sexual cells can survive. Moreover, in animal species, the motility of the sperm absolutely requires the presence of water. In most aquatic animals and some earth animals like the frog, fertilization takes place in the surrounding aquatic environment. In all these animals, fertilization is said to be external because the individuals release their sperm and ova directly into the water. In strictly terrestrial animals, such as spiders, reptiles, birds, and mammals, fertilization is internal. It occurs inside the body of the female. The ovum is thus protected, and the sperm is preserved in a liquid that is ejaculated into the female genital system. In several cases of internal fertilization, in birds for instance, the fertilized ovum produces an egg that will be laid. The embryo then develops outside the mother's body. In these animals, which are called oviparous, the yolk of the egg and the albumin surrounding it are the embryo's food source. Other animals, such as the boa constrictor, are ovoviviparous. In these organisms, the fertilization is internal and the fertilized egg stays inside the female's reproductive system. However, other than the physical protection it provides, the female's body in no way contributes to the development of the embryo. The latter finds its food material inside the egg. Finally, the females deliver completely formed offspring. In viviparous animals, such as mammals, fertilization is internal. The fertilized ova develop inside the mother. Furthermore, the embryos benefit from nutritional contributions from their mother. At birth, the young look like miniature adults. While asexual reproduction allows for the production of an enormous quantity of descendants in a very short time, the advantage of sexuality is clear. It assures better chances of survival of the species. In humans, sexuality is not necessarily related to a desire to procreate. It is wrong to believe that children are always wanted. In countries where living conditions are difficult, contraception is used as a means to keep the population explosion down. On the other hand, in rich nations, parents like to plan the birth of their children for when it best suits them, according to their financial situation or their career. There are numerous means of contraception. Here are a few of them. Couples today generally plan the birth of a child. Most use some form of contraception to delay or prevent a pregnancy. And sometimes, as a last resort, to reverse conception, they resort to abortion. The contraceptive methods used today act mainly by preventing ovulation or fertilization, two prerequisites for the formation of an embryo. The first phase is ovulation, which is the maturing of an ovum in the ovary. The cells surrounding the ovum being formed secrete sexual hormones called estrogen. Through the bloodstream, the estrogen reaches the hypophysis, or pituitary gland, at the base of the brain. In high concentrations, estrogen stimulates the hypophysis, causing it to release a hormone that induces ovulation. That is the expulsion of the ovum from the ovary. Following ovulation, the cellular membrane that held the ovum begins to secrete a new sexual hormone, progesterone. Progesterone prepares the uterus to receive the fertilized egg. As for the ovum, it enters a fallopian tube where it will encounter the sperm. During fertilization, the sperm attempts to penetrate the membrane covering the ovum. A single sperm cell manages to penetrate to the innermost layer. The two reproductive cells then fuse their membranes. Once fertilized, the egg begins to divide. It reaches the uterus after the third day, where it stays to develop during the next 9 months. An oral contraceptive, more commonly known as the pill, prevents ovulation. Oral contraceptives are synthetic substitutes for estrogen and progesterone. Administered in different concentrations than those normally encountered in a woman's sexual cycle, the synthetic hormones disturb the hypophysis, which can no longer trigger ovulation. In addition, the pill makes the inner walls of the uterus inappropriate for the implantation of a fertilized egg. The mucus at the entrance of the uterus is also altered and slows down the sperm's entry. More recently, another female contraceptive that acts on the hormonal level took the market by storm. This oral contraceptive has the power to counter the action of the progesterone. In other words, it prevents the mucus covering the inner walls of the uterus from preparing itself adequately for a pregnancy. Moreover, it is combined with an injection of prostaglandins, hormones that cause the uterus to contract and thereby expel the implanted egg. Due to its abortive action, this contraceptive has raised a heated ethical debate. Indeed, the contraceptive works after the egg has been fertilized. There are still very few contraceptive methods for men. Perhaps that is why there is a great deal of interest in the research into male contraception. It was by taking a closer look at the interaction between the sperm and ovum that researchers first paved the way toward the development of a male contraceptive vaccine. The researchers first identified in a number of animal species a protein interwoven in the membrane covering the heads of sperm. That protein specifically recognizes a molecule called glycoprotein, which is located on the surface of the egg's inner membrane. The fertilization of an ovum is the result of the interaction between the sperm's protein and the egg's glycoprotein. Produced in the testes, sperm acquire that special protein when they proceed through a long, thin, and crooked tube called the vas deferens. The contraceptive vaccine therefore consists of injecting this protein, isolated and purified, into the male. Once it has reached the bloodstream, the protein is recognized by the immune system as a foreign body. The immune system then proceeds to produce antibodies against the intruder. The antibodies reach the vas deferens and attach themselves to the sperm's membrane, opposite the protein. Concealed by the antibodies, the protein no longer has access to the ovum's glycoprotein to fertilize it. The effect of the vaccination is, of course, reversible. The protein is gradually degraded, and the antibodies disappear as well. A protein similar to that in hamsters is being tested on human sperm. If successful, it will be possible to use that human protein not only in men, but also in women. The same vaccine injected in women would probably produce antibodies as well, which would spread through the female genital system. Here, they would attack the sperm present after intercourse. With the development of such highly refined, reliable, and easy-to-use contraceptives, couples will be able to have a much more satisfying sex life. And children will be conceived when they are most wanted. While some couples plan their families, there are others who are unable to conceive. In North America, it is estimated that 15% of couples in their reproductive years face problems of infertility. The first test tube baby was born in England in 1978. The birth of little Louise Brown encouraged many other couples to try in vitro fertilization. Today, thousands of children the world over were conceived this way. In recent years, researchers have noted with some concern a deterioration in the reproductive capacity and quality of man. This deterioration is in part attributed to the fact that couples decide to procreate at an age when the semen has suffered the effects of time. Stress and prolonged exposure to such various environmental pollutants as lead, mercury, cadmium, plastic products, and solvents no doubt also contribute to degrading the quality of human semen. Today, medical science offers many means of circumventing fertility problems. the most revolutionary of which is in vitro fertilization. When a couple faced with fertility problems consults a specialist, the latter first attempts to identify the causes of the infertility. The woman is examined to make sure her ovulation is taking place normally, and to determine whether her cervical phlegm, a viscous fluid secreted at the neck of the womb, is slowing down the motility of the sperm. The inner genital organs are viewed using different methods to make sure the fallopian tubes are not blocked. Sexually transmitted diseases sometimes cause serious infections in the fallopian tubes that can later prevent the sperm from fertilizing the ovum. It is equally important that the man be examined as well, since in 40% of the cases of infertility, the problem lies in an abnormality of the sperm. A sperm sample is taken and analyzed in the laboratory for sperm count, quality, and motility. When the obstacle to pregnancy has to do with the cervix or abnormalities in the sperm, the first option is artificial insemination. First, a large quantity of the man's sperm is collected. Only the sperm that manage to go through several layers of viscosity are chosen. They are then placed in the woman's uterus. However, if it is the man who is sterile or not producing any sperm, a donor's sperm may be used for the insemination. As a rule, a donor is selected whose blood type is compatible with that of the woman. Naturally, men suffering from hereditary or sexually transmitted diseases are not eligible as donors. Sperm from the donor is gathered and kept frozen in a sperm bank for a 6-month period. This precaution is taken to reduce the chances of AIDS or any other sexually transmitted disease from being passed on. This enables the physician to follow the donor for a long enough time before the insemination to make sure he doesn't become HIV positive. In cases when the woman's body does not allow for fertilization due to an obstruction in the fallopian tubes or to the production of antibodies against the sperm, in vitro fertilization offers a last hope. In vitro fertilization consists of artificially provoking the meeting of the ovum and sperm in a test tube. Through an intensive hormone treatment, a dozen or so ova in the woman's ovary are forced to maturity. When the ova are mature, the gynecologist takes samples using a needle entered through the vaginal tract and guided by echography. Concurrently, the sperm supplied by the man is treated to isolate the best specimens. Then, in a fluid conducive to fertilization, an ovum and roughly 50,000 sperm are put together. If fertilization occurs, some of the resulting embryos are transferred deep into the woman's womb. The embryos are formed of 2 to 8 cell clusters. To enhance the chances of pregnancy, 3 or 4 embryos are placed in the uterus. This method may, however, lead to multiple pregnancies. 1 woman out of 5 having undertaken in vitro fertilization will give birth to twins. Triplets are much more rare. Because it is now possible to freeze the embryos resulting from in vitro fertilization, the specialists can now reduce the number of embryos implanted in the womb. Freezing the embryos offers the couples another chance should their first in vitro attempt fail. Although in vitro fertilization circumvents a number of obstacles, it does require healthy reproductive cells. At present, researchers are trying to make fertilization possible even in cases where the sperm has been deprived of its motility, as well as its capacity to penetrate the ovum. They are attempting to inject a sperm cell directly into an ovum by micromanipulation. once again, science will probably manage to compensate for nature's deficiencies. Since the beginning of time, people have been interested in foods said to stimulate passion in their sexual partners. Honey, tomatoes, oysters, hot peppers. Scientifically, one thing is for sure. These aphrodisiacs are only as effective as our power of suggestion. It all happens up here.